Okay, let's get started. Today's study tip says, after reading a page in the book, close the book. Then from memory, write a few sentences and keywords about the reading. So this is a good one because we're going to have reading quizzes. We have about 10 or 11 of them in this class. I have you buy the book because I want you to use the book. And so the reading quiz, I have the book next to me when I write them. I'm looking for, so I'm just scanning it because I know it already, right? But I'm looking for bold words, I'm looking for topic sentences, I'm looking for the stories. A lot of students don't even read the stories that are in little boxes along the way. I have to write multiple choice questions, so I'm thinking, is there a list of things here? Could I make this into a multiple choice question? You could make some flashcards with key terms that you're learning. Remember to study them from back to front as well as front to back. You could highlight some things that you want to review. You could have a friend quiz you on the reading. I bet you can do this too. I can read an entire page of your physiology book and not know a single thing that I just read. Like, I can think about something else while I read the book. That's pure talent, right? I'm sure you can do that too. Like, you read the whole page and you go, you know what? I don't even know what I just read. I was thinking about something else while I was reading these words. Okay, so be sure that you're picking a good time of the day when you're alert and awake. Don't read in bed. Oh my gosh, this is not like a fun book. Okay, don't read in bed. Sit in an upright position with a fluorescent light like our classroom. Okay. So this is still, I'm expecting a review for you, chapters 3 and 4. So I'm going to breeze through them. Chapter 5 I also consider to be review material. Part of chapter 6 I think you should already know. By the time we get to 7, it's going to be new stuff. That'll be neurophysiology. So you should have had this in general bio, and if you've taken micro, I'm sure you have this in anatomy. So this will be easy, which is kind of nice as we start. So inside the cell, remember the goo inside is called the cytosol or the cytoplasm, that contains mostly water, a lot of proteins, organelles. Some of them are specialized to contain fat or glycogen. So remember, fat tissue is called adipose. The glycogen, I think I mentioned this last time, 2 places where you can find it. Do you remember? The liver and skeletal muscle. Okay. Feel free, by the way, when I ask you questions like this, to talk out loud. If your family's sitting around watching you, it's awkward, or you're in the lab, but getting yourself involved in these lectures, even though you're not with me right now, is very important. So when I say pause it, be sure to pause it. If I ask you questions and I delay like that, answer out loud so it gets you involved in it. Okay? The cytoskeleton, let's break down that word. Cyto means what? Cell. And skeleton is, like the bones of the body is a skeleton. It's the framework. So the cytoskeleton gives the cell structure, and it's how all those things stay in place, all the organelles. When I was a student, we thought it was when I was in high school, we thought it was just like a water balloon. And no one actually believed that it could be a water balloon, like really how can everything stay in place? But they hadn't really defined the cytoskeleton. And everyone knew it couldn't just be a free-floating goo with nothing organized. It's very organized. Whenever you look inside a microscope, you can see it's organized. So now we know it's a cytoskeleton. That is a bunch of proteins that give structure to the cells. So in this picture, I want you to see the cytoskeleton. It's all these bars kind of going across here. It's like, the frame of a house, right? Or it looks kind of like branches of a tree holding the leaves in place. In this case, we have the mitochondrion, the endoplasmic reticulum, microtubules, ribosomes, nucleus, and the plasma membrane in the corner. So this is what happens if you take a specialized fluorescent dye and you have it attached to the proteins of the cells, of the cytoskeletal proteins. And these are microtubules, so you can see it kind of like a fine network here, like almost like a woven spider web. So the 2 major elements of the cytoskeleton are microtubules, which are the largest. They also give structure to cilia and flagella. And microfilaments, which are the smaller ones, they also give structure to parts of the cells. They are responsible for microvilli. Now, what's the difference between cilia and microvilli? One of them moves, one of them does not. So the cilia, they move, and where would you find them? Can you think of places in the body? I'm thinking of the respiratory system, like the trachea. Cilia moves mucus, trap particles out of the trachea. And where do you find one place to find the flagella? That's in the sperm, right, to help them swim. So those move. The microtubules do not move. So the purpose of the microtubules, micro means small, villi means fingers, so the small fingers there's microscopic fingers, okay? They are on the small intestine to increase the surface area for absorption of nutrients. Okay, so we'll review that when we get to the small intestine. And actin-myosin or myofilaments are microfilaments found in muscle. We'll review those. We're going to do a lot on actin-myosin for muscles. This is a little test to see if you remember all the organelles. So maybe pause this for a second. See if you can label any of these things. I know it's hard to see. They're small and they're all gray, right? But see if you can remember them. And I want you to not only remember the names, but the definitions. Okay, so press pause and see if you can fill these in. Okay, so these are the Golgi, nuclear envelope, mitochondrion, chromatin, blah, blah, blah. You know all these words, I'm sure. Agranular, by the way, means smooth endoplasmic reticulum, whereas granular means rough. So you can call it, we don't usually call it agranular and granular, but it's smooth and rough. Now, I'm not going to make you label these on the exam. So I won't show you a cell and have you name the parts, but I will expect you to know all of the names of the organelles and their functions. So names and functions of all these things here. Okay, you can find the functions in your textbook if you don't remember what they do. Alright, now I'm moving on from this because I feel like you've seen this so many times. Plasma membrane I do want to focus on. Just a reminder that most of it is phospholipids. So here in the picture, all the little circles here are the phospholipids with the strings hanging down. We'll review the structure of the phospholipid. The other part is the yellow part here in the corners. That's cholesterol. And the third part are proteins. So the big purple things are proteins. Some of them have markers on them that are made of carbohydrates. Some of them don't have them, but a lot of them do. So we'll talk about the purpose of those proteins and where they're located. Okay? So first, the plasma membrane structure. Remember we talked about triglycerides. So if you take a triglyceride, you chop off one of the fatty acids, you then add a phosphate group and a non-lipid group with a bunch of C's and H's. Then you're going to change this whole thing now so that some of it likes water, because remember before, the triglyceride, it did not like water, right? So what's the word that we use for that? To fear water is hydrophobic, right? But now that you've added this orange part at the top here, we now have some of it that likes the water, which is hydrophilic. Let me ask you another just side question here. We reviewed this last time. Here's the glycerol, right? Here's the fatty acids. Which of these fatty acids, the left one or the right one, is saturated? And which is unsaturated? Just to review those words. Which one is saturated and which one is unsaturated? So this one on the right is saturated, which means every carbon has its hydrogens, 2 hydrogens, but the one on the left is unsaturated. Remember how I told you it kind of curves out to the side, right? Or sticks out to the side. Alright, and these were the oil, and this was solid like butter. So the phospholipids, the head is the top part here. They call it the head, they call these the tails. The heads are hydrophilic, they face outward and they love the water. 2 fatty acid tails are hydrophobic, they face inward and fear the water. So if you look at the chemistry or the molecular structure here, like we were just showing, C's and H's, there's the P, the phosphate group, and here's the tails. You can see how you could draw it like a cartoon with a big blob circle and then these squiggle lines. Okay, so that's how we draw it. It's not really a circle, though. It's real atoms. Now, if you put lipids together in water, they naturally do this, where they all come together with the tails and then the little heads kind of surrounding them so that they're protected from these water molecules. That's what these are supposed to be. They look like little Mickey Mouse heads, don't they? This is called a micelle. Let's see if I can write the word down. Pointer. Let's see if I can use the pen. This is a micelle. M-I-C-E-L-L-E, micelle. So the micelle is a naturally forming. Notice that the phospholipid in the membrane has a bilayer. So they get together. Let's go back to that. So the tails are together, right? There's the tails right there. And then the heads are facing outward. So they protect each other by making a phospholipid bilayer, unlike the micelle, which protects each other by turning inward. Okay. Second is the cholesterol. Cholesterol is mostly hydrophobic. So I showed you the chemical structure of that last time. It's mostly hydrophobic. There's a little OH group which sticks in with the heads. So therefore, when we see that picture of the membrane, the cholesterol, it looks kind of like a little kernel of corn, doesn't it? You know, the little yellow there. Sits mostly with the tails, but the little teeny part sits with the heads. And the purpose of it is to stabilize the membrane. Keep it, those lipids actually float around. So it's not like a water balloon where it's just staying. The edges actually move around, the proteins move around. Speaking of proteins, remember that proteins are amino acids connected by peptide bonds, right? They are hydrophobic and hydrophilic. There are amino acids that are hydrophobic that want to stay with the tails. So that's how these proteins stay in the membrane. There's parts of those proteins that are hydrophilic and want to stay on the outside of the membrane, parts that are hydrophobic to stay in. So they are not tied down to the cytoskeleton. Instead, they are free to roam. And the reason that they stay in the membrane is just simply hydrophobic and hydrophilic reasons. Just like phospholipid bilayer stays a bilayer because of hydrophobic and hydrophilic reasons. And then we mentioned the carbohydrates. So the carbohydrates are markers. They might mark a type of tissue. They might mark identity with a person, all kinds of things. Okay? We'll talk about proteins more as receptors and all kinds of things as we get into it. So let's review protein synthesis. Now remember, the making of proteins is done with two steps. The first step is reading of DNA and making mRNA. Tell me again. mRNA stands for messenger, right? Messenger RNA. So that step is called transcription. Next, the mRNA escapes from the nucleus, goes out to find a ribosome in the cytoplasm. The ribosome reads the mRNA and makes a protein by putting together amino acids. So this is my picture. You're not going to find this in the book, okay? So this is called translation. And if you have trouble remembering the word transcription and translation, I always remember that when you transcribe something, like a court transcriptionist writes down everything that somebody said. So it's word for word. So this is the language of DNA and the language of RNA are bases, right? A, T, C, and G in DNA. And there is an exception for RNA, right? It's A, U, C, and G. But they're nucleotides. When you translate, you go from different languages, right? So this is going from language of RNA to the language of amino acids. So the language of RNA is A, U, C, and G, but the language of proteins is tyrosine, lysine, tryptophan. Those are all amino acids, and so it's a different language. So that's how I remember transcription versus translation. Okay, let me show you an animation from, this is from the biology textbook that we use for our biology majors. bio 4A and 4B. And what I want to show you is translation and transcription and the animation so that you can kind of see things in action.
The first step in protein synthesis is transcription of RNA from DNA.
This is a CD-ROM that they use in the teacher's book. So I'll show it to you now. You can probably find it online or just watch it again if you like these things. So this is transcription.
The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA. Once RNA polymerase has gone past a sequence called the terminator at the end of the transcription unit, the enzyme releases the completed RNA and detaches from the DNA.
Okay, let me show you translation.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. Here's how translation occurs. As an mRNA strand slides through a ribosome, triplets of RNA bases spell out the amino acid sequence of a polypeptide. It is the job of transfer RNA molecules to match RNA bases with the correct amino acids. Click on each component shown here to find out more about them.
Okay, so I expect you already remember mRNA, ribosomes, tRNA, right?
We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
So your book shows you diagrams of how transcription and translation work. I showed you the animation, but it's a little more detailed than I need you to know. So let's write down the steps of what I'd like you to know, okay? And on the exam, if you write this, you'll be fine. You can draw pictures if you want, but having the basic steps that I'm going to show you, it will be plenty. So first of all, transcription. It's the reading of DNA to make RNA. It happens in the nucleus. Remember, the RNA polymerase came up to the DNA, binds to the region called the promoter. Then the DNA unwinds. We read, I write T here, but you need to impress me on the test and not just write T. Okay, I need you to write thymine, because you know all the words. We did this in lab together. Remember, we reviewed it, so I expect you to know thymine, adenine, but when you read the A, you're going to grab a U, G, C, C, G. When you reach the terminator, you stop. The messenger RNA is completed. It goes to the cytoplasm and meets the ribosome. This is how your book shows translation. So the animations are nice, and now you can use YouTube, to find all kinds of neat animations. So translation is the reading of RNA to make protein. First of all, there's the initiation complex that forms. So we saw that in the animation. The small ribosomal subunit comes up to the mRNA. The first transfer RNA comes. It always has an methionine. Kind of interesting. Then the large ribosomal subunit comes. I don't need you to know the E site, the P site, all that stuff. I do need you to know that it reads the next set of codon of 3, right? There's 3 amino acids that it reads. 3 nucleotides that it reads, and then it brings in a tRNA to match it. The second one then has a matching anticodon with an amino acid, and they form a peptide bond. Then the ribosome moves all the way until you reach a stop codon. So he did mention in the animation the 3 different stop codons. I don't need you to know those, just that there is a stop codon, and then there's a release factor that comes in. By the way, that's a protein. Comes in with breaking everything apart. Now, the messenger RNA goes off to be destroyed unless we need more of it. Okay, the tRNAs just are floating around. The ribosome breaks apart, and the protein has to be modified though, because not every protein wants to have methionine as the first one. So it goes off to the Golgi apparatus where it's going to be modified. Okay, so this is what I need you to know. I don't think there's an 8, is there? Oh yeah, protein is finished. So the trick is then how to memorize these things. Okay, so you're going to find this class, there's a lot of steps. There's a step 1, 2, 3 through 8. There's a step this. Physiology is how things work, right? So step 1, this happens, and this happens, and this happens. And so you can read these over and over again, but the exam is not reading, right? The exam is writing. We're writing for 2 hours to show me what you know. So really the best way to practice these things is by writing them. So think of all the different ways you can memorize these. Okay, you might do a flashcard with each step one at a time. Not a big flashcard, but little flashcards with each step. You might read them over and over. You might have someone quiz you, but you always have to practice putting out what you know, because that's what the test is. The test is putting out what you know. The other thing here is the exam has short answer questions from lecture, and those short answer questions are usually the things I spend a lot of time on. There were 10 to 12 points, and I just spent a lot of time doing translation and transcription. So that's assigned to you, that those topics will be the ones that you need to know for the short answer questions. Okay, so it helps you kind of to focus. If you see me spend a lot of time on a topic and there's a bunch of steps to go with it, you can bet that that will be one of the things you'll find on the test. Okay? Now the remaining chapter 3 topics, we'll review meiosis and mitosis in lab together. We'll also do the RNA DNA replication in lab. So we're moving on to chapter 4 already. So we're at enzymes, which is also part of one of the labs you're going to do. Just a reminder, what is an enzyme? It is a protein. It catalyzes a reaction, which means it helps the reaction go faster. So it increases the rate of the reaction. It works with what we call the lock and key model. So the enzyme acts as a lock and the substrate acts as the key. So I'll show you a picture of that in a second. This, if you take two reactants in the box here at the top, A and B, and you make them into two products, C and D, the reaction will happen faster if there's an enzyme. The enzyme is not a part of the reaction. It's not part of the reactants. It's a helper, but it doesn't actually get used up or chopped up or changed in any way. So in this case we have substrate A, it's green, substrate B, it's purple, pink, what is this, red, and here the big blue cloud here is supposed to be the enzyme. So A comes into its binding site, B comes into its binding site. Remember, this enzyme is a protein, right? Got a perfect fit here. So it acts as the lock, and substrate A and B come in as the keys. Then there's a reaction between A and B to make products, C and D, but look at how the enzyme did not change. So it stays the same shape, same structure that it was before. Okay, so that's called the lock and key model. Third, the names of an enzyme usually end in ASE. So we had lactase that chops up lactose. In lab we're looking at lactate dehydrogenase. An ACE, usually catalase is the other one we're using in lab. That ends in an ASE. That's usually a sign of what it does. So there's a lactate dehydrogenase. You can also tell from the name about what it does. So lactate dehydrogenase, dehydrogen, so to remove hydrogen from lactate or lactic acid. You can also diagnose a disease or a condition that someone might have with enzymes found in their blood. So creatine phosphokinase is one that if someone's not feeling well and they go into the hospital, they do a blood check for enzymes. They can see liver enzymes, all kinds of things. One of the ones they're looking for is creatine phosphokinase. And when CPK is found in high levels in the blood, that means somebody just had a heart attack. Okay, so the higher the levels, the more recent the heart attack. Some people have heart attacks just feel like they have the flu. They don't feel well, they want to just rest in bed. They don't, the symptoms of the pain in the left shoulder, they don't get that. They just feel sick, feel like they have a fever. They lay down and they don't even know that they have had a heart attack. So this diagnosis is kind of easy if you check enzymes. The other thing we're going to review in lab is that enzymes have an optimal pH and temperature. So every pH is important because, remember, the proteins denature, right? So we reviewed this already. They change their structure if they don't have the right pH. And the temperature is very important. So temperature of most of these enzymes in your body, 37 degrees, right? Body temperature. So if the temperature becomes too hot or too cold, enzymes aren't going to be happy. Which is perfect if you have something like a bacterial enzyme and you're eating your bread on the counter, right? If you put the bread in the fridge, then it's going to be, or a freezer even, those enzymes from the bacteria won't be able to work, which is good. It saves your bread from getting moldy. Number 6, assisted by coenzymes and cofactors. So cofactors are metal ions like Mg. What's Mg? Magnesium, right? Calcium would be another one we'd see. And coenzymes are derived from vitamins. So yes, the vitamins you eat every day in your food. You don't have to take vitamins if you eat them, right? Those are coenzymes. So we need vitamins and ions, metal ions in our food so that we can make the enzymes work better. Okay, the last topic for chapter 4 is bioenergetics. It's the flow of energy in living systems. So do you remember these words? Endergonic reactions require the energy input. And what's the opposite? Exergonic reactions. They release energy. Energy is made by the enzyme called ATP synthase. There's again the enzyme ending in ASE. What organelle has ATP synthase? What organelle has ATP synthase? That's the mitochondria. So we're going to review this next time. ADP plus Inorganic phosphate gives you ATP. The reactions that we have that are important for bioenergetics are oxidation reduction reactions. We call them redox reactions. To gain a hydrogen is to be reduced, or you could say to gain an electron, because when you gain an electron, the hydrogen goes with it. Or to donate the hydrogen is to be oxidized, or you can lose the hydrogen, lose the electron to be oxidized. So if you have a molecule that gains hydrogens, we say that is reduced, and if it donates hydrogens, we say it's oxidized. Some people play a little game here with the, let's see if I can find my pointer. I've heard some professors use this one with students. It's called oil rig. Have you heard of this one? Oil rig, R-I-G. So the oil is oxidize, is lose. So to oxidize is to lose the electron or lose the hydrogens. And the rig is reduce, is gain. There you go. Oil rig is a pump out in the ocean to move oil around. So you can oxidize is lose, reduce is to gain. Games like this to remember stuff are awesome. So whatever you got, whatever trick it is, share it with your neighbor. Tell them how you remember this stuff. I try to tell you how I remember stuff, because something will stick with you. Okay. So this is an example of a reaction. It's reversible. This is part of one of the 8 steps of the citric acid cycle. And I don't need you to memorize this step, but what I want you to see is that in this picture we're taking succinic acid on the left and we're converting it into fumaric acid on the right. And with the succinic acid we're going to put FAD, which is a cofactor, okay, a vitamin really. FAD and succinic acid are converted into FADH2. Wait, what did I just say? Yeah. FAD becomes FADH2. Succinic acid becomes fumaric acid. So let's do the reaction from left to right. Okay, let's go left to right. If you go left to right, I want you to ask the question, what is oxidized when you go left to right? And what is reduced when you go left to right? And what atoms are being removed? Okay, so I want you to pause and think about what is oxidized and what is reduced. Okay, when you answer this question, you only get to choose from the reactants on the left. Okay, when you say what is oxidized and what is reduced, you get to choose the word succinic acid or the word FAD. You don't get to choose from the products. Okay, that's one clue. So when we say what is oxidized, remember oxidize is to lose, right? So in this case, who's losing, succinic acid or FAD? As you go from left to right, the succinic acid is losing, right? It's losing the hydrogens to FAD. FAD is taking them away. So we say that succinic acid is being oxidized into fumaric acid. And FAD, it got the hydrogens, right, to become FADH2. So we say FAD is reduced to become FADH2. Okay, so what is oxidized? It's succinic acid. What is reduced? FAD. And what are the atoms that are being removed? This is the easy one, right? Hydrogens. So now let's try going from right to left, because remember the reaction is reversible. Fumaric acid, pretend you're going right to left. Fumaric acid going to succinic acid, and FADH2 going to FAD. So now going from right to left, answer the same 3 questions. What is oxidized? What is reduced? And what atoms are being removed? Pause it and think about it. Okay, what is oxidized? To oxidize is to lose. So who's losing? Now remember, you have to choose from the reactant. So you're going to choose either FADH2 or fumaric acid when we go right to left. Okay, so who's losing when you go right to left? The FADH2 is losing, right? It lost them to become FAD. Who's gaining? Fumaric acid. Fumaric acid, therefore, is reduced to become succinic acid. So in this case, I'll try to write the answers here on the left side. So what is oxidized? Because oxidize is to lose, right? So the answer is F. I need my pen here. FADH2. And what is reduced? Because reduced is to gain, right? So fumaric acid, gain them. Fumaric acid gain. And what hydrogens are removed? Same thing. Or sorry, what atoms are removed? Hydrogens. Same thing. Okay, wrap your head around this. Look at the book for some more things, and then bring me questions if you're still having trouble with it. We're all done.